Uh, our sub-team is upflow aerobic / blanket reactor. My name is Mohammed Aden.
I am Emily Wood.
And I'm Erica Rowan.
And I'm Katrina Chen.
Our research goal is to design a UASB reactor and then treat wastewater and then discharge to downstream and also produce the biogas. So what is wastewater? Wastewater is any water that has been contaminated by human use. So where does it come from? Wastewater sources include domestic, industrial, commercial, and our target is at domestic level. What does it contain? Wastewater contain variety of pollutants and nutrients such as like nitrogen, phosphorus, pathogen, and other microorganisms. Why is wastewater a problem? They can cause pollution, eutrophication, and disease. Wastewater is a problem because untreated wastewater have a great impact on the environment. For example, water pollutant and organic matter at high concentration can kill aquatic organisms like fish and many other organisms. Additionally, nutrients in wastewater cause eutrophication, which cause algal bloom, leading to the reduction of oxygen concentration, especially in the deeper layer. This makes it hard for aquatic animals to survive. Untreated wastewater also affect human health and drinking water source, which can cause the spread of some very nasty waterborne disease such as cholera. Next slide. This is a UASB conventional design. As you see, the wastewater enters from the bottom and flows up. As you see, blue arrow. When the water enters, the reaction starts. And bacteria breaks down organic matter and produce biogas, mostly methane and carbon dioxide and other traces gas. This UASB this reactor works in absence of oxygen. So this you can on the top you can collect the gas, and that gas you can use for cooking or lighting. And then the blue arrow leafing is clarified when you clean the water, that clean water goes to downstream, but still they have a microorganisms, but they are better than we. And then bottom red arrow is the sludge, like biosolid. You can use fertilizer or other variables. Next slide. This is AguaClara UASB design. So this reactor is a single tank. Wastewater enters and flows upward through the suspending sludge blanket made up anaerobic microorganisms. Blanket filter clean the wastewater by removing organic contaminants and pathogens. Wastewater traveling from the peristaltic pump enters to the UASB reactor through the holding tank at the top. As you see, the water is going down. But the pulses follow reactor water collecting in the tipping bucket until it reaches volume predetermined by the placement of pivot on the side of the blanket. When it's filled, the blanket tips, sending the pulses wastewater down to the pulse pipe approximately every 15 minutes. A clear, so, and this reactor cleans the water and produces, after cleaning water, then discharge to downstream. And then also we collected the gas by connecting the biogas bag, and that gas they can use for cooking and lighting, similar like conventional. So I want to pass my colleague Emily. And
Thanks, Mohammed. So to give a little context for our current work this semester, last spring, in spring 2020, we had just finished installing a temperature control system for the UASB reactor when the COVID-19 pandemic hit. And due to the pandemic and construction at the wastewater plant, We were forced to halt our research and disassemble the UASB reactor for the time being. And this was incredibly disappointing, but one sign of encouragement for our research was the discovery of healthy sludge granules during this process. Our reactors are currently being stored at the Ithaca Area Wastewater Treatment Facility, and the sludge granules are kept in cardboard containers, as you can see on the right, and are fed occasionally with wastewater to maintain their viability. We're hoping to regain access to the wastewater plant next semester, fall 2021, in order to continue our research. Until then, we're focusing on two main goals. The first is documentation of UASB design and literature, and the second is lab experiments to improve the biogas collection system. The UASB team has been doing wastewater research since 2013, which means that new members have to sift through 8 years of reports, presentations, and literature reviews to fully understand why the UASB reactor was designed the way it was. And so one of our main goals for this semester was to document 3 main aspects of our research to aid in the transfer of knowledge between semesters. And those aspects were how water flows through the UASB reactor, the history of design decisions, and the third was literature that described the biogas production rates.
So upon discussion with Dr. Richardson, we wrote a function to calculate the theoretical gas production in a day. The gas flow function, shown above, is used to calculate the biogas production rate given the average influent rate, the COD load, and the temperature. There are assumptions from literature, mainly from a source called anaerobic reactions. For the specific estimated calculation done with Dr. Richardson, we used an approximate influent rate of 300 liters per day, which is calculated from the height and retention rate of reactor. By molar ratio from the reaction shown above, the function will calculate the amount of biogas for the COD load input. The volume that the gas occupies can then be calculated with the temperature with the ideal gas law. The pressure is assumed to be standard pressure since the pressure will vary much less than the temperature, and any change can be neglected since it is very minimal compared to the other factors from the reactor. We then use this function to calculate the biogas production rate for gray water and black water. For gray water, the function calculated that an influent rate of 300 liters per day, 500 milligrams of COD per liter of wastewater, and at standard room temperature 25 Celsius, around 57.34 liters of biogas is produced per day, which is roughly the amount of gas that we calculated by hand. For black water, each liter of black water has approximately 2,206 milligrams per liter of COD, which we found through literature. With the same influent rate and temperature, the function calculates that the black water will produce 253 liters of biogas per day. And now Katrina will talk a little bit about our biogas collection system.
Like Emily mentioned, one of our team's main focus this semester was to tackle the gas collection system so we can better understand it and prepare for the reinstallation of our reactor next semester. Our current gas collection system consists of a tube coming out the top of our reactor that is connected to a wet test meter as seen in the figure on the left. The gas then passes through it and is collected in a 60-liter gas bag. The wet test meter will indicate the volume of biogas produced because the dials turn as the gas passes through it. In spring 2020, our reactor was still in the startup phase, and we saw little to no biogas production being recorded. We were curious to see if we were actually producing no biogas, or if our wet test meter is not sensitive enough to report small amounts of gas due to low pressure. Another possibility is that there could have been a leak in our gas collection system. To tackle these problems, we plan to run experiments in the lab with 2 different gas counters. One is the wet test meter and the other is this MilliGascounter. The MilliGascounter is filled with a packing fluid and counts gas bubbles to digitally report how much gas has passed through it. It is more accurate for smaller flow rates of gas. However, the end goal is not to switch to the MilliGascounter as it is very pricey, but this may be necessary if the wet test meter cannot accurately measure the small biogas flow rates expected during the startup period of our reactor. We wanted to test the sensitivities of both gas counters to compare them to each other. To do so, we set up an experiment in lab with a peristaltic pump so we can control the flow rate. The figure on the left shows our actual lab setup, and the figure on the right is a schematic of the lab setup that more clearly shows the gas's path. A fixed amount of time was selected to run the pump, and calculations were done to see how much gas we're expecting to read on the devices. This value would then be compared to what the device actually portrayed. To see if low pressure is affecting the accuracy of the wet test meter, we vary the flow rates of air being pumped in. For each of the flow rates, we plan to do identical experiments on both devices to see if and how their readings differ. To test for any leaks, we are using soapy water at any of the joints to make sure our system is airtight. For more detailed lab notes and data, you can check out our appendix slides. Here you can see our data for the wet test meter. We started at 40 milliliters per minute since that was a calculated flow rate from the biogas calculations Erica discussed, and worked down from there. For the majority of the flow rates, we did 2 trials to test the precision of the wet test meter, as seen in the table. The values of the 2 trials differed a maximum of 0.02 liters. These results indicate that the wet test meter is precise even at lower flow rates. We also evaluated the accuracy of the wet test meter by comparing the wet test meter reading to the expected values. When comparing these numbers, the differences varied from 0.01 to 0.04 liters. The inaccuracies are likely due to error of the tubing or the peristaltic pump itself. Overall, because we do not need to know the exact volumes of biogas produced, the wet test meter should be precise and accurate enough at low flow rates for our purposes.
As previously mentioned by Katrina, one of the ways we considered to increase the precision of biogas reading was to use the Ritter MilliGascounter machine. However, when we were reading the manual, we realized that the packing fluid for different gases were different. Due to the susceptibility to change with CO2 based on its solubility and acidity, it is best to use a dilution of HCl. But when the manufacturer of the counter was contacted, she informed us that because the MilliGascounter was not calibrated with HCl for our specific model, we cannot use it with HCl, which means that we will not be able to accurately test the amount of gas flow within the system. Since our initial reason was to increase precision and the reading from the instrument would not be precise anyways, we decided to take out MilliGascounter as part of our lab experiment. And as Katrina mentioned, the wet test meter was accurate enough. Another problem we encountered was the overflow of the manometer on the wet test meter, which means that a high pressure buildup within which means there is a high pressure buildup within the wet test meter. During this time, the hands on the meter were not counting. We fixed this by lowering the flow rate. Since the wet test meter and the MilliGascounter are for lab tests only and are not required to be attached to the reactor, these challenges should not impede our reactor in any way. However, there still is a problem of gas escapage, which we hope to resolve in the future.
So our first priority after we regain access to the Ithaca Area Wastewater Treatment Facility is to restart our reactors. It's important to do this as quickly as possible because the time it takes to generate sludge granules and come to stable biogas production rates is on the order of weeks and months. And the end goal is to eventually compare the performance of pulsed flow UASB reactors to continuous flow UASB reactors. Reactor performance is measured by comparing the effluent organic matter content in both the quantity and quality of biogas. So organic matter content in wastewater is typically measured using a biological oxygen demand or a chemical oxygen demand test. Based on this semester's lab experiments, we will be measuring biogas production rates using the wet test meter, while the quality of biogas will be measured using a gas chromatograph. A gas chromatograph is able to measure the relative percentages of methane, carbon dioxide, and other gases present. And for our situation, we ideally want a high percentage of methane in our biogas to maximize its efficiency and usefulness as an energy source. So thank you for listening to our presentation. We welcome any questions and recommendations, and you can reach any of us at our emails below. For more information, you can see this link about expected biogas calculations or these links with additional information about our laboratory experiments.
Great, thank you.